Hi, my name is Amber Boats, and I'll be presenting our study on the early life origins of brain aging, looking at cognitive and brain aging in the Dutch famine birth cohort study. To quickly introduce myself, I'm a PhD student within the Amsterdam University Medical Centers in the group, the DARE group, Developmental and Reproductive Epidemiology. And being within the session after these previous talks, I don't think I still need to introduce that factors in early life can drastically alter development, or in other words, your genes are not your destiny. And we see this all throughout biology, for instance, in plants, in insects, and in animals. And for instance, these dandelions, their soil that they grow in and develop in their environment has a huge impact on what they will look like. And how tall they will grow or how many leaves they will produce. So their environment has a massive impact on how they develop. And it's no different for humans. The environment that we grow and develop in has a huge impact on our health throughout life. And especially during the first period of development where we grow and develop the most and the fastest, which we usually define as the first 1,000 days of life, from conception until your second birthday, is the period where you're also most vulnerable for these environmental exposures. And that also holds for the brain, because actually most of the brain develops before birth, and therefore it's very sensitive for environmental adversities such as nutrition and stress, especially during this early development. And prenatal and early life circumstances can also affect late life brain health. Or in other words, what happens in the womb can last a lifetime. And actually, these early life factors, these exposures during your early development may also impact late life brain health, and may also be part of the factors that determine why some individuals achieve normal and healthy brain aging, whereas others may develop symptoms of dementia and late life pathological brain disease. We recently published a systematic review where we studied the association between prenatal exposures and brain volumes in brain regions that are associated with Alzheimer's disease, where we clearly saw a pattern throughout literature of smaller brain volumes in these regions, which is also why we really feel we need to stress that Alzheimer's disease should actually be viewed from a lifespan perspective. If these brain regions that are essential for late life brain health are not able to develop to their full potential, it may have an impact for your late life disease risk. And that brings me to our study of the Dutch famine birth cohort study, where we look at long-term effects of prenatal undernutrition. And I'll quickly introduce the historical background. So the Dutch famine is a period at the end of the Second World War. It was the winter of 1944-1945, where as a result of war circumstances in the Netherlands, there was a brief period of a very severe famine of a few months. And this famine was mostly very harshly experienced within the large cities, including Amsterdam. And the famine occurred all throughout society, and also pregnant women experienced this famine. And the famine also ended pretty suddenly at the end of the Second World War. After the Netherlands were liberated, there were, for instance, food droppings, and just the availability of food altogether increased very rapidly. And 50 years after this period of famine, we identified a group of birth records of individuals that were born around the time of this famine within the hospital in Amsterdam. And we retraced these individuals, and they have been followed ever since. And those are the individuals that are included in the Dutch famine birth cohort study. And they have been followed now for nearly 30 years. And in our cohort, we include 5 study groups. We have 2 control groups that were born before the famine and conceived after the famine. And we usually combine them in our analyses as a single unexposed group, because we keep seeing that they are very similar in their outcomes. And then we have 3 exposure groups of individuals being exposed during late, mid, and early gestation. So because this famine only lasted a few months, and we selected our individuals based on birth date, we can very clearly identify during which trimester these individuals were exposed. So trying to summarize 25 years of research into a single slide, it looks something like this. And there's two things that I would like to stress out based on this slide. The first thing is that there's clearly a lot of late life health effects that we see associated to prenatal exposure to famine. And the second is that we mostly see these effects in the group that was exposed during early gestation, which is very interesting because that's the group that actually seemed healthy at birth. They had normal birth weight, but these are the individuals that experienced the most effects of their prenatal undernutrition on later life health outcomes. And because we see the most effects in this group, the results I'll discuss later are mostly focused on this specific exposure group. And at the age of 68, it was the first time that we started looking at their brains, and especially of this early exposure group. We saw that prenatal undernutrition affects brain structure and cognition. And we also see that it's associated with accelerated brain aging, looking at a brain age gap estimation paradigm. We see that it affects brain perfusion. And we've recently analyzed the resting state data, seeing that we saw that it affects late life resting state brain functional connectivity throughout the brain. So taken together, these results indicate that there may be a pattern of accelerated brain aging after prenatal undernutrition. However, brain aging obviously is a longitudinal process, and this was just a cross-sectional study. So everything that we speculate about brain aging, we cannot know for sure. It may also be effects that have been present since early development. So most of my research project has been focused on performing a longitudinal follow-up study of this brain imaging study at age 74, where we try to identify which changes in brain structure and function we can observe over time. So how do these individuals age? How do their brains age? So I already said, last year we published our results on resting state functional connectivity at age 68. We looked at within and between network functional connectivity of the default mode network, salience network, and central executive network, where we saw that there were sex-specific effects on both within and between network functional connectivity in different regions of these resting state networks. So we're currently working on a longitudinal analysis combining the data from age 68 and age 74, looking at changes in resting state functional connectivity over time for within and between network functional connectivity of these three networks. And we're using latent change score modeling to look at change over time, which is a form of structural equation modeling. And we have already run the resting state analyses, but we're currently working on the structural equation modeling. So if you have any experience with this and any ideas and thoughts, I would love for you to share those with me. So please come and find me if you have any ideas. And then the second study that we're currently working on that I'd like to introduce is the Stroop task study. So the first time that our participants performed the Stroop task was at age 58. And the Stroop task is a very well-known cognitive task that measures cognitive processing, selective attention, and response inhibition. And the assignment is pretty easy. You have color words which are printed in an incongruent color ink, and instead of having your first response of reading the word, you need to suppress this response and instead name the color of the ink that the word is printed in. And at age 58, we saw that exposed individuals made more mistakes and they had a slower response. So that made us wonder what's happening inside their brain while they're performing this task. So during the last imaging data collection round, we redesigned the Stroop task to be able to perform it as an fMRI task. We made it a bit easier because we saw last time it was introduced as a stress task, and it was very, very hard. So individuals were making so many mistakes that it's hard to measure a brain response of Stroop task performance instead of just measuring a stress response. So we made it a lot easier to enable most of our participants to be able to actually perform the task correctly. And we also introduced a control paradigm where they had to name a colored square to correct for all kinds of scanner distortion. And actually, most of our participants were very capable of performing this task correctly. So that was good, considering the fact that these were 75-year-old individuals within an MRI scanner. And this is what our overall pattern of brain activity during the Stroop task looked like. And it's actually very similar to the pattern that we see in the neurosynth meta-analysis of the Stroop task. So that makes us very confident that we've actually measured a Stroop task response. And in our study, we decided to focus on a few regions of interest which have been associated with Stroop task performance, and also specifically with Stroop task performance in brain aging and mild cognitive impairment. These are the dorsolateral prefrontal cortex, the posterior parietal cortex, and the anterior cingulate cortex. And we're currently running these analyses, so these are preliminary results. But we observed no statistically significant group differences. But there were a few interesting points that we observed. First of all, we saw that our control groups were actually no longer similar. We've observed throughout the years a pattern that our control groups were very similar, and we were able to combine them into a single control group. But in this case, the pattern seems to have shifted, where actually the born before control group seems very similar to the early exposed control group, and the conceived after control group is the group that's different from the other two. We actually don't really know how to explain this, apart from the fact that we clearly now observe that the exposed group is more similar to the older group. And also we see a lot of spread in all our data points, but especially in the conceived after group. So if you have any thoughts on this, you'd like to share with me, please come and find me, because I would be very interested. So thank you for your attention, and I'd be happy to answer any questions.